Our next speaker, Steve Petrou from the Florey Lab, has a lofty title. He works in research with Ingrid Scheffer. He's head of the Florey, the ion channels labs, and basically does a lot of this extremely remarkable science in dishes with mice. He's going to tell you some exciting things, but I just wanted to say a couple of things because, as you can see, I like my introductions to be a bit informal. Steve is one of those people who's making stuff happen. Anybody who signed up got an opportunity to have a tour of Steve's lab, and that was very exciting. You got an opportunity to see what he's doing. Steve runs tours like that for some big corporate bigwigs here to try to get them to give more money so that we can have the research done. And this is just a call to action, folks, because if you guys know wealthy people in the world, you call them out, right? So Steve will give those folks tours of his labs. Some of those people are worth a lot of money. This is not a poor country, right? And these will be people who are worth millions. And they'll walk into Steve's lab and they'll say, this is incredible. You guys, this is amazing work. You're right, groundbreaking. And I'm going to pull out my checkbook. I'm going to give you $5,000. Wow, really? Let's just think about our bills, I don't know, for the month. So, this is my one little moment to say we have got to call it out, everybody. If you know people, we have examples of folks here who are great fundraisers. I am often told as an American, you guys are just better at it over there. How come? No, not necessarily. I know a ton of generous Australians. So I would like to encourage all of us to encourage our Australian and international friends to be even more generous. I mean, what are you going to do when you're dead, right? You can't spend it then, so get them to give. Get them to give to people like Ingrid and Anne and Steve and Gavin and all the great researchers. So there you go. That's mine. And now I'm going to bring Steve Petrou to the front. Welcome, everybody.
Thanks, Sarah. So what I'd like to do today is talk a little bit about some of the therapeutic choices that are being considered for rare neurogenetic disorders. It's a really, it's an amazing time now because so many things have converged. Our understanding of the genetics, our clinical knowledge, technology to enable to screen, and we saw a beautiful example from Gavin's lab about how technological advances are allowing us to measure the function of the proteins and the genes we're interested in remarkable ways. Our choices for therapeutic modalities, and I'll obviously get into that more during the talk, are there. But importantly, there's a whole bunch of things that are converging to try and drive this. And this is the scheme I use in my lab and in some of the commercial programs that I'm involved in. As you would have seen from the first slide, I'm involved heavily in 2 companies where I'm running their science programs to help really deliver therapies in the rare neurogenetic space. Obviously there's urgent clinical need for everybody in this room and for their kids. The more we understand about the genetics, the more we understand about disease mechanism, the more we know that we've got a technological approach for screening and for finding drugs, and that we've got the appropriate models in place such that we can bring these things together. And where we're at now with respect to commercial engagement, it's at least in America now, and even in Australia, you would have heard last week about the government reimbursing for Spinraza, and that's for a rare neurogenetic disorder, spinal muscular atrophy, and it's a remarkable time. In America, there's additional incentives that are created by the FDA there, which is the Drug Approval Authority, that lets rare programs get additional benefits with respect to patent life, with respect to special vouchers that these small companies that do this can get and can sell on to larger companies to help defray the costs of developing drugs. So it's a really good time to try and do something, and that's why we're working so hard to try and make something happen in the therapeutic space. And this is sort of an overview of, we've got severe forms of epilepsy that emerge early in life. We've got comorbidities. As Ingrid said and others, can't forget about the comorbidities. And the question is, are there therapeutic decisions we can make about our designing criteria that will address not just the seizures that we all talk about, but all the other really important issues. And this sort of just reiterates all the things that I said. So we're just trying to drive this model as hard as we can. We're trying to take on as many genes as we can. And it's great to have people like Gavin here because we can bring vertical expertise in things like SYNGAP. I mean, we've got some vertical expertise in our lab, but we can't be, at that level for all the genes that we work in. But we think we might have some therapeutic directions that we want to go in that can leverage the expertise of all the international and other workers in this area. This is the list of, just a partial list of the genes that have been found in early epileptic encephalopathies and other neurogenetic disorders. And there's our goal, really, to develop this very deep understanding of the pathology, make the models, understand from those models what's required of a therapy. Do we need to turn the gene off? Do we need to turn the gene on? Do we need to do something else to the gene? Is there an additional point of sensitivity that we can exploit to help us give us an edge that help us modify this disease process? We really do want to modify the disease process. And disease modification means you're not just treating the symptoms. I mean, all current or most current anti-epileptic drugs really focus on stopping seizures, but all the underlying causes and many of the other things that give rise to the seizures aren't addressed. And that's particularly so in the epileptic encephalopathies and in these rare neurogenetic disorders that we've been talking about today. And we really want to just change the course of the disorder. This is just a snapshot of some of the genes that we've got in our lab at various levels of activity. Probably on the top are the ones that we're putting most of our effort into, but we've got models and we're working on programs that touch on most of these. So it's quite a big lab, and we're doing a lot of things, and we're trying to really intersect these genes with the therapeutic modalities that are on this slide. Antisense oligos, and I'm going to talk a lot about that in the thing. Repurposing, and you saw that from Gavin. Small molecules, that's something that we're driving in our commercial programs. I mean, taking large chemical libraries and then doing medicinal chemistry on those with chemists is a very, very expensive process. And often that's better suited for commercial development where you can put 10 or 20 or 30 chemists onto the molecule to do the necessary modifications to turn it into a tool compound, into an actual drug that you can take, and that drug's not metabolized too quickly, and that drug gets to the right spot that it needs to get to in the brain, and that drug doesn't cause any toxicity, either in the short or long term. There's a lot of hurdles in getting a small molecule into an actual patient and having it safely there. And then there's a whole bunch of regulatory and other conditions you need to meet in order to actually make it a thing that you can buy. And that's where I think the commercial partnerships come in very handy, because that's what they're really good at. Peptide toxins is a program we started with Glenn King. He'll talk about that, and I'll leave it to him, but there's a whole other wealth of molecules in that peptide toxin basket that act, we all get bitten by snakes and spiders and creatures in the ocean, and they hurt and they paralyse and they stop us from breathing. So they're very biologically active, and that's how they've evolved. And Glenn's doing remarkable work to try and understand, are there alternative uses for these otherwise toxic and venomous compounds. So what I want to focus on today is something that's very near and dear to our lab, and that's this so-called RNA-based precision medicine. And why do we like RNA? So I'll do 101 molecular biology. The central dogma of molecular biology is this idea that the DNA at the top is the blueprint of all of us. This is what's in our cells. This is what makes us, who we are. Now that DNA is converted into an intermediary. So the DNA is double-stranded. There's normally two of them, but it's converted into that pink intermediary that's called messenger RNA. And that's the actual blueprint that the body uses to make the proteins that assemble us. So it doesn't make it directly from the DNA. It makes a copy from the DNA. That copy is then rearranged and manipulated in millions of different ways. Bits are cut out, bits are lopped off, other bits are exchanged. It's heavily modified, it's influenced by other factors, and then the final blueprint's available, and then the proteins, as it's like a string of pearls, are added according to that codon. So each of those 3 letters says put on, in this case, tryptophan, phenylalanine, glycine, serine. These are the proteins, the amino acids that you hear about when you know that's in all our food and supplements. It's like a string of pearls. They get linearly arranged, and then those amino acids fold up into a little machine that becomes the protein that does the work. And all different ways of folding that gives the proteins all the myriad of different functions that they have. So this is the central dogma, and you can see that the RNA in the middle is right in there. So it's a nice available target for manipulation. And if you look at a mutation, how does a mutation move from DNA to RNA to the protein? You can see here if there's a... Is that up? Oops. I'll just point. At the top, that star, is there might be a change there, one change. And it only takes one change to make a kid sick. And that one change changes the DNA, that changes one of those codons, which is that little 3-letter code that tells you what amino acid to add, and that might change this phenylalanine to some other amino acid. There's like 20 amino acids. If it changes it from phenylalanine to anything else, it can disrupt that protein and produce the change in function that gives rise to the disorder. It's just a tiny little thing when you look at it, one tiny chemical change. And we're asking the question, and Anne Paduri talked about CRISPR, how it might go up to the DNA and actually fix that mutation in a person permanently. And there's a lot of hope that that will work, and there's a lot of challenges still. And I'll touch on that very briefly at the end. But we're asking the question, well, now that we've got this RNA and it's there, what can we do to RNA to try and fix the disease? And luckily, because there's a lot of ways to break a gene. You can see here, you can mutate the protein, get increased function of the protein, reduce function. You can actually, as I said before, the way the protein is put together from these little building blocks before the messenger RNA is finally processed, it's like a big chunks of it interspaced with these things called introns, and you might take this exon and put them together, and how you do that actually affects the function of the protein. That's called splicing. There's other regulators of the protein. And as Ingrid pointed out, you can get large-scale deletions and insertions or chromosomal rearrangements that can also contribute to the disease. So there's many ways to break our genes to then break our proteins and cause disease. And we're focusing on RNA because there's one common technology called antisense oligos, which is an artificial piece of DNA. It's a small piece, 18 to 20 DNA molecules long. It's chemically modified so that it's stable in your body. Most DNA, if you just took it, wouldn't hang around, but you chemically modify these molecules and they can stay around for months. And then you can use these in all these ways to try and manipulate your RNA. And one really good example of this is Spinraza. Some of you may have heard it, this drug that was released last year, and it's for spinal muscular atrophy. And this is quite clever actually, because there are 2 versions of the gene that gets mutated that causes spinal muscular atrophy. SMN1 and SMN2. Normally, the number 1 version of the gene makes the normal protein that you want. Now, and then version 2 of the protein essentially is non-functional. We don't know what the regulatory reason for that is, but it's non-functional because of the retention. This is what I mentioned before about how we can piece together the RNA. This piece and this piece, all the little bits in the middle get removed and you end up with this concatenated string. Normally in the SMN2 gene, number 7 is just gone for some reason, and it makes the resulting product non-functional. Now that normally doesn't matter because you've got SMN1 doing its thing. But if you get a mutation in SM1, you can no longer have the function you need, and that leads to these muscle and motor neuron deficits. And the idea in this therapy was, well, can we activate this silent 2, version 2 of the protein? And that was done by making an antisense oligo that's either called nusinersen or Spinraza that forces the retention of this particular part of the RNA. And once they did that, and they developed the molecule, that was a collaboration. It started at Cold Spring Harbor, and a collaboration between a company called Ionis and Biogen. It's been released as a molecule. It went through all the safety testing, all the necessary things, and these kids are getting remarkable efficacy now. So much so that the Australian government supports the reimbursement of this molecule to the tune of $250 million a year. It's an expensive treatment, but the government's stepping up. And it's the only reason the government stepped up is because parent groups were, standing, behind the health minister at the time he made the announcement. And it's got to come from parent groups, that grassroots support, if we want to move these things from ideas into actual medications. And that's great. And now I think the field is poised. There's actually, one of the companies we started is operating in this space for SCN2A. And there's all companies dotting around the world now that are starting to take advantage of this technology to look for therapies for these rare disorders, because we can be very, very targeted with RNA. We can make the RNA specifically interact with any part of the genome we want. It's not random, it's very, very specific. So I can almost guarantee that we can get efficacy, that we can manipulate the RNA we want to do. The risk comes, can you make those molecules safe? And can you formulate them in a way that you can use them as a drug? But that sort of removes a big element of risk that you can face with small molecule programs. And this is just an example of I mentioned the different ways that you can break genes and RNA. Well, in this case, if you have a mutation that has gain of function, so there's more protein activity than you actually want, a really simple use of the antisense oligos is you can make an antisense oligo attached to that RNA and it'll kill it. And there's natural mechanisms in the body when they detect that antisense molecule is bound to the RNA, will degrade it and remove it, and you'll get less protein. And that's a very obvious and one of the first uses of this technology clinically. And I won't go into the details of the mechanism, but suffice it to say that it's relatively straightforward to reduce RNA levels. And as I said, the challenge is, can we do it in a way that's safe, that we don't have to... These molecules need to be introduced through the spinal tap, so intrathecal delivery. You can't take them orally and get them into the brain. The good news is that you only need 2 or 3 doses of this per year. And so there's going to, and the centres are starting to get specialised to be able to do this, and it's going to become more and more routine. And I was talking to my wife about this. Well, 90% of pregnant women have this when they get a spinal block, when they, for pregnancy. So it is a common method. And I think, most parents that I've spoken to, if it means that they're going to get efficacy and child's health's going to improve, it's something that's very tolerable. Now, there are other tricky ways of manipulating. What if you want to increase the function of what if you don't have enough? So I'm showing that as a tiny little protein here. Rather than a big lot, you don't have a normal amount, you don't have enough. And fortunately, there are several options available for increasing the level of the protein. I talked about these antisense molecules, these artificially introduced ones. Well, the body's got natural ones. For, about 40% of our genes have got natural antisense molecules that are hanging around there and are inhibiting the normal levels of protein, and it's one of the body's natural ways of controlling how much RNA is made. So the idea is, well, if you've got a natural molecule that's blocking and you don't have enough protein, what if you block the inhibitor? So you block the blocker. And by blocking the blocker, you remove its ability to reduce the level of that protein. So you use exactly the same technology. An antisense oligo that binds to the blocker. You take the blocker out of the equation, you get more RNA, you get more protein. This is something that's been actively tried in Dravet syndrome. There's a company in Texas called OPCO who's doing what's so-called natural antisense inhibition to try and increase the levels of SCN1A protein in Dravet. SYNGAP1, there's a potential for SYNGAP1. There is a natural antisense in the human for SYNGAP1. That's something we want to explore, and we've been talking to Gavin about that, and obviously with parents involved, with Danny and Danielle about, is this a viable option and can we get this off the ground? So something we're very interested in to do. Now, in addition to those methods, there are other ways you can do it. you can increase the expression of a protein by, this is way overly complicated, but RNA has got the blueprints in here, and prior to the blueprints, there's some instructions on how to use them up the front of them. And so normally the system looks at the instructions and it says, well, do this, do that, and then I'll go ahead and turn that RNA plan into a protein. Now those instructions up front, you can manipulate those in a way so you get more RNA made again. And there are options there for SCN2A, for instance. We're looking at this particular approach. Again, bind an antisense oligo to a critical part of that instruction sequence, and you can increase the amount of RNA. Some proteins are a candidate for this, some RNAs, others aren't. In some cases, the instructions are cut off before you get an opportunity to bind them, so you can't use it. And that's why we're exploring all the myriad of ways that you can manipulate RNA, because if you have 3 or 4 ways, chances are you'll have at least one way for the RNA of interest in your particular disorder. So we're very hopeful here, and this has been proof of concept work has been done in other genes, showing that you can get big increases in that. An additional problem, what we heard about that today, was the so-called dominant negative effect. And that's essentially the concept, the protein that contains the mutation is like the bad apple, and it makes the other apple in the fruit basket get mouldy as well. That's called dominant negativity. And often, for therapy, it might be enough just to remove that bad apple. One apple's enough to have normal function, but the fact that the protein that's got the mutation is influencing the healthy copy makes the disease way, way worse than it otherwise would be. So are there ways, and I'm showing that here by the mutant copy with the wild type normal protein gives the function just of the mutant. Now, how can we specifically target? Well, you can think, well, there's a mutation, let's make a drug that specifically targets that mutation. The trouble with that is that there are hundreds of mutations in different patients across the world, and it's not possible to develop an individual antisense oligo for every single individual patient. So that gets you thinking, and then you say, well, hang on, maybe there are other ways of achieving the same thing. And all of us have got variation in our genomes. They're called SNPs, single nucleotide polymorphisms. And there's just hundreds in some genes, and they're variation, natural variation that sometimes changes the protein, sometimes doesn't, because there's redundancy in the genetic code. Different signals can still give you the same protein. So these SNPs now, and because you see in the population some SNPs are very common, there's 60% of people have this, and 20% have this other one, and 10% have this other one, you can think of a strategy that says rather than binding the actual new mutation that's going to be unique and private to every patient, can we attach an antisense oligo to that SNP that's on the same gene chromosome as the mutation? And therefore, if you make 2 or 3 ASOs, you can then say, well, I can now target the ASO to the allele that's linked to where the mutation is. There'll be some cases where people have exactly the same SNP on both chromosomes. They won't be candidates, but more often than not, because you get one gene from your mum, one from your dad, you do get variation that you may be able to take advantage of. And that's something I'll show you like in this slide. The mutation is here, and because this is a private mutation, but we can make an ASO to the fact that there's a common difference here in different patients. You can see the colour sequence here because of this change in the nucleotide. So this ASO will work for 60% of the patients that have got a mutation. So this is one way of overcoming that when you need the so-called allele specific. The other way of doing that is if a mutation appears frequently in the patient population. For some genes like DNM1, there are mutations that are very common. And that means you can actually specifically target just that mutation because it covers 40% or so of the population. And that's a program that we're working with a company in California called Ionis on to try and see if we can get something done in that space in an allele-specific way. So the final way, I know this is a lot to take in, but what I want to give you a flavour for is that there are a lot of options based on this RNA mechanism. And so the final one I'll talk about is yet another regulatory mechanism. Normally, as I said before, these intervening stretches of DNA are discarded and the two green bits come together like here. But in some cases, so the bits that make the protein, the jargon is exon, the bits that don't make the protein are called introns. And if this intron is not removed, it's called retained. It's a retained intron. And that's a natural mechanism that the body has for naturally reducing RNA expression. We see that in cancer cells. We see that in the body, that this is a natural way. Now, if you go back to what I mentioned with nusinersen Spinraza for spinal muscular atrophy, you can use an ASO to direct splicing. So in the same way that we could make an exon come and go, we can use an ASO to remove this red bit. And as soon as we remove that, we now get 10 times as much of this RNA as we otherwise would have. And this can then bolster up the flagging amount of RNA, and that's something we're thinking about for SYNGAP. I know there's a company in Boston called Stoke Therapeutics that's doing this for Dravet and SCN1A. We are exploring this for our SCN2A program, and I'll mention the reason why we're going up and down for 2A in a minute. So there's enormous wealth. So we're sort of focusing on this therapeutic modality. Not that other modalities are worse, but we can't do everything. And in our research, we can focus on this. There's a huge common chemistry, which makes it more tractable. We learn how to handle and deliver and design ASOs. But by using them in these subtly different ways, we can affect the systems very differently. We can turn genes on, turn genes off. We can turn exons on and off. We can even ask the question, are there other genes that we can regulate that might help the disorder? So use the body's natural repertoire of proteins, manipulate those in creative ways that may give you improved disease outcomes. So one of the things here, I won't go into them in detail, but it just gives you a flavour for all the different mechanisms that we can target with these ASOs. And so I'm very excited by the potential of this. We're putting a huge amount of effort into this process, and we hope to start delivering. I think for SCN2A, there'll be an announcement from one of our companies in a few months that for a major partnership and clinical trials, maybe by the end of next year, we'll be very close to starting our first trials. So we're very excited and once we do that, it sort of helps open the floodgates a little bit for trying to get more interest, more support into these programs. So once you've shown you've done it. Spinraza was important. Every Antisense oligo talk I go to now, everybody starts with the Spinraza story. Now look at this, it worked. It's a very exciting thing. Just to put that into context, this has largely been driven by one individual, by a guy called Stanley Crook, who started the company. It was originally called ISIS, for obvious reasons. They changed their name to Ionis about 5 years ago. But he's been at this for over 30 years. Working tirelessly for these, and pushing really hard on these rare disorders. And it's just people like him are remarkable in the effort and the single-mindedness. And only now the field is starting to catch up. All the other things that need to happen to make this a success are finally happening, and that's why I wish them all the success in the world in their programs. Now, coupled to this is that we need to get really smart about which patient gets which drug. Even if you've got a mutation in SCN2A, for instance, how do you know that you're a candidate? Because when we look at 2A, we see mutations that increase the function, and we see mutations that decrease the function. And certainly, if you've already got reduced function in 2A, you wouldn't want to take an ASO that reduces that further. And also, if you have a mutation that increases function, you wouldn't want to take an ASO that increases the activity of that protein. So in parallel to these efforts to develop the therapies, we're looking at a companion diagnostic. So once we have knowledge of the mutation, can we use that to predict what the actual mutation is going to do functionally, and then direct it to the right therapy? And this is just one approach we've taken here. It's actually, we use a very standard method, which is we can make the mutation, put it into a cell line rather cheaply and rather quickly, but but we need to get that channel to be operating like a neuron. So we combine it with a computer model in real time, and we can take the one protein and it has the behaviour of a collective of 7 or 8 proteins working by merging it in this model in real time. And what it lets us do, this is the normal protein in an individual who doesn't have a mutation. This is the protein function in people that have this loss of function, 853Q. Clinically, these kids present differently. And you can see it's loss of function. This is bifidus mutation, gain of function. This is early onset epileptic encephalopathy with 1882Q, gain of function. When you do this assay across a range of stimulations for the nerve, this is actually what a nerve would look like. If you were to put an electrode in a nerve in the brain, this is what you would record. They're called action potentials. So it's a direct correlate of what we would see to be excitability. And then we can look at these curves, and we can say this is normal in black, loss of function in blue, and curiously, the bifidus, which is a self-resolving inheritable form of SCN2A, so this is something that can be passed on, but work by Sam and Ingrid and others have shown that this, the kids deal with this themselves, is similar. It's similar in some ways to this encephalopathy that you can't actually naturally control, but it's got this difference down here. Under low levels of stimulation, the encephalopathy mutation is way more active than the bifidus one. And that sort of tells us, we can separate. It also tells us that maybe a therapy that just pushes this down a bit is going to be sufficient to make it look like bifidus, and the patient can naturally handle that. So they're very useful tests. We've taken these same cells from patients and made stem cells that Snezana was talking about. We get exactly the same pattern for the excitatory mutation. So this cheap and dirty assay that we can do in a few weeks, and it's not going to cost a fortune, and we can do it in the timelines that matter for the delivery of the therapy, in it looking like a good predictor. And with similar work now is being undertaken for Dravet mutations, GLUT1 mutations, and we're thinking about these companion diagnostics. And I think we've got to get that together with our our discovery, together with natural history studies. All these things need to come together so that we can quickly move from idea into delivery of a therapy. And the one last thing I'll talk about is we mentioned this CRISPR technology. And one of the concerns, of course, with CRISPR is that in your attempt to try and fix the mutation here, there's a lot of liability that molecule, the same process that fixes it, might introduce random mutations elsewhere in the genome. And that's permanent. And you worry about that until the technologies become perfect. You fix the mutation, but you give the person cancer or some other disorder. And that's a concern, and it's not quite ready yet to be so specific. But there's a different form of it called Cas13 that acts on the RNA, so rather than the DNA. And as I said before, the RNA is like a temporary photocopy of the DNA. If you fix it in the RNA, you should have the same effect on the protein, but if you then don't have a permanent expression of this CRISPR system, you're not permanently changing the genome, and you might have the same impact in a safer way. And I know there's groups now, we're not working on this, but there's groups around the world who are looking at CRISPR in RNA as a safer alternative, and maybe something that can be delivered earlier, and then like 10 years after that, 20 years after that, maybe CRISPR for the DNA, maybe to get to the point where it can be safely delivered. I'll leave it there. Lots of people involved. This is my group. I've highlighted Chris and Snezana, who are obviously very senior people and run their own programs as well. Really, really important part of the team. Big collection of people, funding, and my commercial interactions. Thank you.
Stay with us. If we have some questions, I'd love to hear them from you. And just while we're waiting, do we have our microphones? Yeah. So if you have a question, raise your hand. Scientists often talk about CRISPR. Does everybody in here know what CRISPR is? Would anybody like to know? Yes. So can you just explain a little bit what CRISPR is?
It's a system that was hijacked from virus and bacteria that was used to manipulate their own genomes. And scientists have hijacked that and have said, you know what, we can turn that activity onto a mammalian genome. So you can actually, this molecule, you put the CRISPR system, you put a bit of artificial nucleic acid that you make, and that finds a region in your gene, let's say where a mutation is, and it can bind there, and it can actually edit, change a nucleotide from an A to a T, and fix the actual mutation that caused the disorder. And that's the hope, and that's the excitement. So it's a way of going in and editing our genomes very specifically. And people are very excited by that, because you can say, well, we can apply this now and fix disorders. All inherited disorders or de novo disorders can now be repaired with this. But there are challenges, like I said, in your effort to fix it there, the system isn't so specific that it may not cause problems elsewhere. And then we've got to ask the question, even in a child's brain that's very large, how do we get this system that's normally delivered by a virus into the entire brain? Works great in a mouse that's tiny. You want to get into a person, you can't do 27 injections to get
That actually leads me to the next question, which is from Jim, KCNQ2 Cure in the United States. What delivery system does Steve have in mind for RNA, virus or injectable?
Yeah, so what the good thing about antisense oligos is that they're naked. You just deliver them into the cerebrospinal fluid through the spinal, and that's got natural circulation, and they just get in. And they self-deliver, so you don't need an additional, which is great. As soon as you introduce viruses, you worry about immune responses and other issues that you have to deal with. They're not insurmountable, but they're just additional challenges that slow things down and make them more expensive.
Other questions from, yes, we have one over here. And then, oh, sorry, you've got that one. That's fine, go right ahead. We'll do that and then Claire.
Just with the RNA research that you're doing, is there other things that are happening? Because I know that we have other issues going on, like celiac disease. Could the RNA research that you're doing sort of lead into other sort of, I guess, revelations in
the disease process?
Yeah.
I mean, the way we're thinking about it, if the celiac issues are a result of that mutation, in addition to the other things, this approach should fix everything. And the caveat I'll give is if you've got to fix it, and we don't know how early back in the child's development do you need to go to make this a complete fix. And we heard from Gavin about that. Maybe if you give it at this age, you'll fix 70% of the things. Some things may have already passed and you can't turn back the handle. We just don't know how much reserve there is. The hope is if you get it in the right time, in the right amount, that all the things, all the different phenotypes, the comorbidities, the intellectual disability, everything should be resolved without us having to think about, because what we do in the past, we say, let's look at epilepsy, we've got epilepsy, we've got a movement disorder, we've got a mood disorder, we've got all these different things, and you do research on those individual things, and it's like that whack-a-mole game, and everything you got, there's 100 things popping up, and you're trying to bang them down. And
that's how parents feel, by the way.
Yeah, I'm sure they do. So can we go back and, to the root cause to stop any of that from occurring, and then we don't have to think about them, I'm too old to think about 27 things.
All right, we have a question over here, I think from Claire.
Thank you. My question was actually related to this, because I was going to ask more around the critical period that Gavin mentioned as well, because obviously it gives so much hope for us, but obviously our kids are, a bit more grown up. So
I think this is always the challenge for parents. As we work hard and as the children get older, you do wonder, when do they pass out of
the range where they, and what we don't know is we really don't know for sure in people when that number is. Chances are that there is going to be something that happens that we can't reverse, all right? And that's just, we'd love to be able to, maybe other therapies might help. If you fix the cause of the disease, maybe there are a few other things you can do to help deal with those, maybe not as comprehensively as the ASO, but in a supportive way. We just don't know the level of plasticity there is. And we're only going to know, that was with Spinraza last year when they started giving the drug to older kids. They saw some benefit they didn't necessarily expect to see. I don't want to give false hope, but also don't want to say it's all doom and gloom, because we really don't know.
Well, and any parent who's tried a new medicine on an older child, and that's medicine, not that, but you can see, whoa, that did something. So I think, yeah.
Right. And if you see, there are some clinical signs, if you see changes in the brain through MRI, those sorts of things might be very hard to reverse.
Yeah. So we have a question up the back, and we have a question over here from Anne. So we'll do Anne, and then we'll come up to the top. We're going to get Dan working around.
Yeah, thank you very much. I'm just curious about the amount of treatments you may need, because I understand on a layman's basis there are millions of cells in the body. So how do we hope to treat all the DNA
Yep.
that's out of alignment?
A lot of these
Number of
genes
treatments?
that we're looking at are only expressed in the brain. So the injections we give will be contained within the brain juice, the CSF. And that'll get to every cell, and because these molecules last so long, they have multiple rounds of destroying thousands and thousands of copies of the RNA that you don't need. So you'll get an injection probably every 3 months or so. And that's what's happening with the Spinraza.
And that resolution will be permanent?
No, you've got to keep giving it because you're acting on the RNA. A new RNA gets made, it's got the old fault from the DNA, and you've got to give it again and again and again.
Steve, what do you think we should do as a scientific community and as a parent community to raise awareness, but also to try to address the issue of scalability? You know, when you have one child getting CSF injections every couple of weeks, every 4 weeks, you know, that it's an endeavor for the families. It's a huge expense that the hospitals, at least in the U.S., are not willing to necessarily bear. When you come up with something, and I think you'll come up with something, how do we scale this and how do we get the system to be ready for it?
So the health economics arguments of this are going to get extremely complicated. It's beyond my ability to fully understand the regulatory and health economics issues that we're going to face. On one hand, if the kids are doing really well, they're presumably not consuming other resources that could be turned towards, you know, intrathecal injection clinics. And that may not become just the province of a tertiary care clinic. You might develop specific clinics for that activity that can inject a whole bunch of these. And, you know, only time will tell. It's going to be, you know, I don't know what that future is. Parent groups, government groups, everyone's going to play a part in finding. I mean, my biggest concern now is that they're quite expensive, these treatments, all right? They can be $700,000, $800,000 a year. The Australian government, US health insurance are covering it. Will they cover it when there are 10 Spinrazas? You know, maybe eventually as things run off patent, you'll get generics. That'll fix the problem. People need to recoup the millions or hundreds of millions of dollars needed to develop these. So I think, you know, the system will sort of self-regulate. It's hard for me to envisage, if there is a drug that's going to treat an SCN2A kid, it's hard to think, well, the system, society as a whole is denying that kid that treatment. And I think we just got to do it and hope that someone else helps us fix it.
Up there.
Oh,
at the top
hi.
right.
This all sounds amazing. Can you hear me? Yes. Is there a ballpark for how long when this is going to be delivered, or like 5, 10, 15,
No, I mean,
20 years?
End of next year, we're doing SCN2A trials. We've got other drugs lined up that'll be, once we get the important proof of concept in our preclinical models, if I can attract a commercial partner to then stump up the $20, $30, $100 million it's going to take that idea and develop, because when I've got a mouse version, it's not the one you're going to put into a person. You've got to develop a human-specific one, and then you've got to make a safe version of it. Not every single version you make is safe. There's a whole bunch of testing. So but it's just a question of running this model. That's why we're looking at so many. I'm doing whatever I can to get as much money and resources as I can to try and put some parallel processing into this.
I want to make a point too with where you're at when it comes to the stage that he's at, and if you have an organization, what is going to help him out and be behind him are the patient group.
Absolutely.
So you need to prime your patient group with what you know in the research, so that when they're ready to launch a clinical trial, you know, in this stage, which is so huge and exciting, you keep your patient group abreast. The word will spread, and then they're going, you know, he's going to be looking for a company. Well, that company is going to want to know that the patients are behind it, because they're not going to invest if they don't know that those patients aren't behind that trial. So that's where patient groups come in, and you start rallying for this. And so that's going to help him find an investor, because they're going to see that active patient group. And so that's my biggest advice. And so that's why I try to keep my patient group abreast of as much as I can, so that when the time comes, it's going to be a lot easier to get there. And that's why patients are so incredibly important.
Couldn't have said it better.
Right, and I think that is applicable whether or not you're SCN2A. I think that this
Absolutely.
advice is applicable incredibly broadly. And again, you know, there's got to be a bit of a Three Musketeers approach, because things are going to go as they go, and we've got to be a bit of all for one and one for all. Thank you so much,
you.
Steve. We really appreciate it. A lot of hope there.